E. coli happens, a presentation on my term project of my favorite microbe. Escherichia coli is a very well-known bacteria, as it is the most studied free-living organism worldwide and has 11 Nobel Prizes associated with it. E. coli lives commensally, beneficially, and pathogenically in human hosts and also in other warm-blooded mammals. E. coli is gram-negative and rod-shaped. The motile strains have flagella at one end and adhesive fimbriae pillae or chemical adhesion covering their whole structure. E. coli's dualistic nature is demonstrated by humans living symbiotically with it, as it helps to synthesize vitamin K, which is an important blood coagulation factor, as well as helping synthesize B vitamins. It also helps us break down our food for easier digestion. E. coli can offer protection to their human host against other harmful microbes. As previously mentioned, E. coli is also the cause of gastrointestinal diseases, as well as causing urinary tract infections, neonatal meningitis, pneumonia, and blood infections that can colonize the lining of the heart, causing inflammation and the destruction of the heart valves, leading to endocarditis. E. coli strains fall into four main groups: Group A, Group B1, Group B2, and Group D. Group A includes most of the commensal and symbiotic strains. Group B2 and Group D include the pathogenic strains that make mammals ill. Of the gram-negative bacteria, E. coli are bacilli, lactose-fermenting, and reproduce quickly, about every 20 minutes. Shigella is another form of bacteria in the Enterobacteriaceae family that causes gastrointestinal symptoms and harms the human tissues from the Shiga toxin, similar to E. coli. Unlike some strains of E. coli, Shigella strains are non-motile. The classification of such closely related bacteria is sometimes difficult to attain through 16S rRNA analysis. E. coli grows and thrives in human hosts and are heterotrophic, meaning they derive their nutrition from an outside source. They are the most common coliform, which are bacteria that function on aerobic respiration and can also be facultative anaerobes that can exist in the absence of oxygen. Coliforms are bacteria that are gram-negative, rod-shaped, and ferment lactose. They reproduce by binary fission, which is a type of asexual reproduction, and as mentioned, can reproduce rapidly, every 20 minutes. Nutritionally, they consume carbon, hydrogen, oxygen, nitrogen, and sulfur. They acquire carbon from the biosynthesis of organic molecules that were ingested by their host. When growing E. coli on a petri dish, glucose is served up as their primary carbon source, and the salts supply nitrogen, phosphorus, and trace metals. They are quite hardy and can survive at a temperature range from 4 degrees Celsius to 53 degrees Celsius. E. coli has interactions with other microorganisms it comes in contact with and can participate in horizontal gene transfer by acquiring genetic information from another microorganism, as well as passing on information. Microorganisms exchange information through the genetic elements such as plasmids, integrons, and transposons. Plasmids are a key player in the development of molecular biotechnology as they deliver the DNA of one organism into another. Seeing that plasmids are circular, they do not dismantle very easily, but they do reassemble easily once the new DNA is added. E. coli is the preferred bacteria to use for a plasmid transfer because it replicates quickly and its anatomy and physiology is well known. E. coli is also widely available. Its ability to reproduce aerobically and anaerobically makes it a perfect host organism in biotechnology. A lot of bacteria can be killed or at least controlled by specific drugs called antibiotics. E. coli has developed resistance to a lot of antibiotic drugs. One type of resistance is the beta-lactam ring, which is a core part of several antibiotics that disrupt the cell wall. Strains can gain resistance by producing extended spectrum beta-lactamase through plasmids and horizontal gene transfer. E. coli is very adaptable and has been shown to persist through harsh environmental conditions. Therefore, as it travels worldwide and between humans and animals, it can spread its resistance. My graphic novel was a story about how I got sick on my trip to India, most likely with traveler's diarrhea, which is also known as enterotoxicity toxigenic E. coli. My story weaved between my journey and E. coli's innocent aspirations to become world famous and make a great impact worldwide. The story concludes with my finally getting rid of my illness and E. coli's dreams coming true. Thank you for watching.
Ladies and gentlemen, today I stand before you to shed some light on the fascinating world of Azospirillum, the superstar of nitrogen-fixing bacteria. Picture this, a gram-negative, slightly curved, and dare I say, a bit swirly-shaped microorganism. Azospirillum living up to its name like a true celebrity, azo for nitrogen fixing and spirillum for its fabulous spiral shape, knows how to make an entrance. But it's not just about looks, my friend. Azospirillum is all about the moves. With its trusty flagella, these bacteria can swim and groove through liquid environments like nobody's business. They're the disco dancers of the bacteria world, shaking their flagella to the rhythm of life. Now, let's talk about their social life. Azospirillum is the life of the party when it comes to microbial interactions. They can rub shoulders with other nitrogen-fixing bacteria like Rhizobium, forming a squad that knows how to fix nitrogen in style. But that's not all. Azospirillum has a secret identity as a plant whisperer. These bacteria can cozy up to plant roots, making themselves at home on the root surface or even sneaking into the intercellular spaces. It's like a symbiotic house party down there. Why is Azospirillum the VIP of the microbial world? Well, my friends, it's all about the nitrogen game. While we're breathing in that lovely 78% nitrogen in the air, it's not the kind of nitrogen we can make use of. But fear not, Azospirillum is here to save the day. With their trusty enzyme nitrogenase, they break that triple bond in nitrogen gas, reducing it to ammonia or ammonium. It's like they're the nitrogen superheroes, converting nitrogen into a form that plants can party with. And boy, do plants love to party with Azospirillum. These bacteria are plant growth promoting maestros, increasing crop yields by a whopping 5% to 30%. That's like throwing the best plant party ever with Azospirillum as the DJ spinning the hits of growth and abundance. So, ladies and gentlemen, let's raise our glasses to Azospirillum, the life of the microbial party. They dance, they fix nitrogen, they make plants grow, and they always the talk of the town. Here's to that bacteria rock star that keep our soil fertile, our plants thriving, and our nitrogen cycle on point. Cheers to Azospirillum.
16-year-old Tatum went to a 4th of July barbecue cookout and ate some chicken wings that were mostly undercooked. His friends ate the chicken too, but because Tatum has severe allergies and a weakened immune system, Tatum contracted severe salmonella poisoning and admitted himself to the ER the morning after when he repetitively started throwing up. While admitted, Tatum experienced stomach bloating and inflammation, diarrhea, nausea, and severe dehydration. Doctors prescribed him ondansetron, an anti-nausea medicine, and started him on fluids for the first day. They knew, based off of Tatum's history and whereabouts, that he had contracted salmonella poisoning, which is a type of bacteria that can cause foodborne illness in humans and animals, and are commonly found in the intestines of animals and can contaminate food products. Since Tatum is immunocompromised due to his allergies and has a hypersensitive immune response to harmless substances in the environment, his microbiota was compromised and could not easily fight off the salmonella contamination. In a healthy individual with a well-balanced microbiome, the presence of beneficial bacteria can help prevent salmonella poisoning. These beneficial bacteria, such as certain strains of E. coli, Bifidobacterium, Lactobacillus rhamnosus, and other commensal organisms, can compete with salmonella for nutrients and attachment sites in the gut. However, when the microbiome is disrupted, such as through antibiotic use, the protective effect of these beneficial bacteria can be diminished. This disruption can create an opportunity for salmonella to colonize and cause infection. It is because of his compromised immune system that doctors collected a stool sample to test what specific strain of salmonella bacteria had colonized Tatum's gut. The genus Salmonella is part of the family Enterobacteriaceae, which includes other well-known bacterial genera such as E. shigella and Yersinia. Within the genus Salmonella, there are two species of primary importance to human health: Salmonella enterica and Salmonella bongeri. Salmonella, Salmonella uh, phylogeny demonstrates that the bacteria have undergone evolutionary diversification, leading to the development of different lineages and serovars with distinct genetic traits and host preferences. The outer structure of Salmonella consists of several layers. It is the cell envelope, which comprises the outer membrane and the peptidoglycan layer. This envelope protects the bacterial cell and helps maintain its shape. Inside the bacterial cell, Salmonella has a cytoplasm containing genetic material, including DNA, as well as various cellular machinery and metabolic structures responsible for its survival, replication, and production of toxins. Furthermore, Salmonella has evolved strategies to survive and thrive within the host's gastrointestinal tract. It possesses violence factors that aid its attachment to the intestinal lining, allowing it to resist the mechanical and immune defenses of the host. Salmonella can also modify the gut environment to its advantage, such as by inducing inflammation or altering the metabolism of the host. Tatum's doctors collected his stool sample and looked at it under the microscope. Dr. Wittick and Dr. Lipp found bacteria that were rod-shaped and gram-negative. Further, the bacteria appeared as elongated rods with a length of approximately 2 to 5 micrometers and a width of around 0.5 micrometers, a hypermotile flagella, heli, fimbriae, surfaces of the intestine, and appendages that adheres to and colonizes the intestine and appear to be a facultative anaerobe, all checking out to be a Salmonella enterica. However, there was one last test to conclude their diagnoses and to proceed with proper treatment. Salmonella enterica exhibits a high degree of serotypic diversity. Serotypes, also known as serovars, are defined based on variations in surface antigens such as the O antigen and the H antigen. Different serotypes of Salmonella enterica have distinct antigenic profiles, allowing further identification. Dr. Lipp performed an agglutation assay of the bacteria collected from the stool sample and identified the surface antigens of Salmonella enterica, including the O antigen and the H antigen. For Tatum, this meant that his immune system may have reduced functionality or be less effective in mounting an immune response against these antigens during the agglutation assay. Dr. Wittick took a blood sample from Tatum and checked his white blood cell count. Increased levels of white blood cells may suggest inflammation or infection in the gastrointestinal tract, and sure enough, Tatum's white blood cell count was elevated. Tatum was quickly started on broad-spectrum antibiotics called fluoroquinolones and given a cocktail of vitamins and mineral supplements to activate his immune system to fight off the pathogen. as well as nutritious foods that are fiber, probiotic, antioxidant, and polyphenol rich to boost his gut microbiome. Doctors explained to Tatum that he had to be monitored only for the next 6 hours to see improvement in his condition because Salmonella enterica maintained an anti antagonistic relationship with the healthy microbiota in his gut, using its virulence factors to colonize and invade host tissues. Tatum was feeling better as he was just feeling nauseous, but he was told his body was getting adjusted to the medication and his microbiome was working extra hard to get rid of the infection. Doctors started to note that Tatum was feeling much better and ready to go home because his inflammation, liver bile fluid activity, digestive processes, and overall condition was showing much improvement. Considering that Salmonella bacteria can tolerate a wide range of pH levels, which the optimal pH for growth and survival would have been 4 to 9. Since Tatum was throwing up and experiencing diarrhea, his stomach was only acidic for a short period as a result of the explosion of stomach contents, including gastric acid. But since he had limited food and nutrient availability, the Salmonella bacteria remained in the gastrointestinal tract's alkaline environment. Once Tatum was given vitamins and these minerals and nutritional food, Tatum's bile fluid activity showed improvement, and the pH was not showing alkalinity as bile contains various antimicrobial substances, including bile acids, phospholipids, and immunoglobulins. These components have direct antimicrobial effects. Further, doctors did explain to Tatum that Salmonella is a mesophilic microorganism that thrives in temperature range known as a danger zone, like several human pathogens, as well as the human microbiome, which is between 40 degrees Fahrenheit and 140 degrees Fahrenheit. This struck Tatum as he was discharged and made a full recovery while being able to enjoy a turkey burger at home. He began to comprehend the causes and contamination sources of salmonella poisoning in food and some solutions and safety hazards to consider. He began to think about the food source, which is the livestock, and its journey to being packaged and sold at grocery stores. There is a great chance that raw meat contains many microorganisms, including harmful bacteria such as salmonella. Tatum began to understand why proper refrigeration, proper cooking, and temperatures are important to prevent the growth of salmonella. Refrigerating raw meat in temperatures 40 degrees Fahrenheit and below inhibits the growth of salmonella as it can not survive in colder temperatures. Correspondingly, cooking raw meat thoroughly in temperatures in 150 degrees Fahrenheit and above kills the bacteria present, including salmonella. The heat penetrates the meat, reaching the core and ensuring that the bacteria are destroyed. Another contamination source that Tatum thought of was linked to water sources. Salmonella can contaminate water sources through various means, leading to waterborne transmission of the bacteria, and properly treated or untreated sewage or wastewater can contain salmonella. Animals, including livestock, poultry, and wildlife, can harbor salmonella in their intestines by ingesting the water. Inadequate sanitation practices during water collection, treatment, or distribution can lead to cross contamination. For example, if equipment or storage tanks are not properly cleaned and sanitized, salmonella from one water source can contaminate other water sources. Employing water filters is a solution to help ensure that you are drinking safe water. Tatum thought all these measures should keep him safe, but he went the extra step to make sure to wear food preparation gloves. By changing gloves and washing hands between handling different types of food, such as raw meats and ready-to-eat foods, you can minimize the transfer of bacteria from one food to another. Tatum feels ready to keep himself and others safe from food poisoning. He is now more cautious to make sure that the food he is eating is fully cooked, does not have an off or unpleasant odor, unusual texture or consistency, which are all signs of food spoilage, and presence of bacteria such as salmonella.
Staphylococcus are gram-positive bacteria that are mostly found on human skin, nares, which is the nasal cavity, creases of the body, and other areas. Staph means cluster, and coccus means spherical. Each staph is about 0.5 to 1 micrometer. Staphylococcus are non-motile, it means they don't move around. They are non-spore forming, and they are facultative anaerobes, meaning they can survive with or without oxygen. Staphylococcus colonization in the human skin is completely normal, but if there's a high level of Staphylococcus in a broken skin, it can cause infection.
There are common three types of Staphylococcus that are found in humans that can cause infections. These are Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. All of these three types are catalase positive. There are ways to differentiate the three types of Staphylococcus. Between these three types, Staphylococcus aureus is the only coagulase positive, and both Staphylococcus epidermidis and Staphylococcus saprophyticus are coagulase negative. Now, to differentiate the two, a novobiocin susceptibility test can be done, which is an antibiotic. If the bacteria is novobiocin sensitive, it is Staphylococcus epidermidis, and if it's novobiocin resistant, it is Staphylococcus saprophyticus. Staphylococcus aureus is usually found in the skin and the nose. It can cause skin conditions such as boils, folliculitis, cellulitis, and impetigo. Staphylococcus aureus also releases toxins. First toxin is hemolysin. This is a pore-forming toxin that destroys erythrocytes. Second, leukotoxin. These attacks the leukocytes and dies from necrosis. This causes the tissues to die and can lead to organ failure. Third, exfoliatin toxin causes staphylococcal scalded skin syndrome. Fourth, enterotoxin. These are generated when Staphylococcus aureus lands on food, and it can still be active even after the bacteria is killed by cooking. So this results in food poisoning. And last, toxic shock syndrome or TSST1. These are produced at the site of the infection that can cause physiologic changes such as fevers, rash, decreased blood pressure, and organ failure that can cause death. Staphylococcus epidermidis is usually acquired in hospital, and it is a common cause of nosocomial blood infection. These infections happens to patients with prosthetic valves, cardiac devices, catheters, and the use of IV drugs. This is due to the exposure of the bacteria while the procedures are being done. Staphylococcus saprophyticus mostly colonizes in the perineum, rectum, urethra, cervix, and GI tract. This bacteria is the common cause of UTI, especially for sexually active females. Skin infected with Staphylococcus can be treated by topical antibiotics. Boils and abscess can be drained by healthcare professional. And if the infection is inside the body, it can be treated with oral antibiotic. And for severe cases, healthcare professional can also administer IV antibiotics to kill the bacteria effectively.
My microorganism is Vibrio harfei, which is a marine bacteria. It's a member of the genus Vibrio in the family Vibrinaceae, order Vibrionalis, class Gamma Proteobacteria, and the phylum Proteobacteria. It's pretty genotypically and phenotypically similar to Vibrio campbelli. It's gram-negative, bioluminescent, rod-shaped, and it has polar flagella to use for its motility. It's a facultative anaerobe and is able to use different carbon sources for its metabolism. It lives in multiple niche environments, which is the reason that it's able to use different carbon sources depending on the availability. And this can be both in a pathogenic symbiotic, or it can also be a planktonic bacteria, actually. Within the squid, it forms a symbiotic relationship, which is what you can see on the screen right now. The bacteria lives inside of the light organ of the squid and provides the bacteria with nutrients in exchange for its bioluminescence, which the squid then can use to attract prey, as well as it uses it also for camouflage in the moonlight as well. And once the bacteria is settled inside of the light organ, it will stay there throughout the lifespan of the squid. So usually bacteria enters during the time where squids are juveniles, and then the rest of its lifespan, they're doing their thing inside of the squid. And when there are not a lot of options for this bacteria to be planktonic or be in any kind of symbiotic relationships, it does become more virulent and searches for a host to be pathogenic, and eventually will kill the host as well. And because of that, it's becoming increasingly more dangerous marine bacteria. It's been around for a long time, but because of global warming, the temperature of the waters are rising, and this bacteria really likes warm water. It started out in tropical waters, but has since spread all across the world. You can find it in areas in Europe now, in Asia, where it's a huge problem as well. And this is a big threat to our food industry. An example of that is the shrimp in Asia. So the bacteria can cause multiple different diseases, including eye disease. It can cause gastroenteritis, and in case of the shrimp, luminous vibriosis. And luminous vibriosis is pretty recognizable since it actually makes the host glow, which is a pretty obvious thing. And that's because the V. harvei is inside of the stomach, and it uses the chitinous lining of the stomach as the carbon source for its metabolism. So that's what's going to make it glow, especially in the abdominal area. And that will kill the shrimp. The shrimp also start swimming in all kinds of different directions. There's just all kinds of really sad things when they are infected with this bacteria. And this is really hurting shrimp farmers as well. So there's a huge demand in the world for shrimp. It's a big industry, and over the years, aquaculture farmers are losing more and more money each year on these different diseases that are plaguing the shrimp specifically, and of course, other industries that are plagued by this bacteria and its relatives as well. So a lot of the times in aquaculture farming, one of the main ways to prevent things like this and to treat the product, I guess, the shrimp or the whatever else I guess you're farming, is using antibiotics. But as we know from lots of different industries, and also just from our world and antibiotics for humans, there's more and more antibiotic resistance that seems to be popping up all over, which is why they're trying to find a different, more creative solution that could be not related to antibiotics, and also just that there's a demand from people for their food to not be raised with antibiotics. So that's another big motivator for that as well. The bacteria never really grows bigger than the size that it starts at inside of the squid, as going with my squid example, I guess. It stays in there, it grows, it bioluminesces, and some of it, as the population grows, some of it does get released into the water, but in general, the major colonies stay inside of the squid.
Hi, this is Iris from microbiology. I chose Rhizobium as my favorite microbe. Nitrogen makes up around 78% of all atmospheric gases. Almost all of that nitrogen exists in its diatomic form, N2. No plants or animals are able to utilize nitrogen directly. The only organism capable of utilizing nitrogen are groups of prokaryotes called diazotrophs. Some plants have evolved to form symbiotic relationships with diazotrophs. The main example of this is legume rhizobium symbiosis. Rhizobia get nutrients from the cells of legume roots and fix atmospheric nitrogen into ammonia, which is used by their plant hosts. Rhizobia that exist in free-living forms in the soil are incapable of nitrogen fixation. However, during symbiosis, Rhizobium undergoes physiological and structural changes that allow it to do so. Bacteria in the genus Rhizobium are rod-shaped. They usually have flagella and are mobile. Once it's in the legume host, Rhizobium transforms into an irregular branch shape. Rhizobia are gram-negative genus of bacteria. Rhizobium's traits of nitrogen fixing and legume association are not phylogenetically significant. Some archaea, cyanobacteria, and several other bacterial groups who are not phylogenetically related to Rhizobium also have the ability to fix nitrogen, which suggests that this trait is a result of convergent evolution. Rhizobium's capacity to associate with legumes is most likely the result of horizontal gene transfer amongst bacteria, as opposed to vertical gene transfer, which is much more phylogenetically significant. The Rhizobia group has been described as both paraphyletic and polyphyletic. The Rhizobium genus splits into 3 subgenuses: Rhizobium, Bradyrhizobium, and Azorhizobium. All 3 fall under the category of proteobacteria, but on distinct branches, each of which also includes bacterial species that are not rhizobia. Rhizobia are heterotrophs. Heterotrophs are organisms that cannot make their own food. Heterotrophs get their nutrients from other sources of organic carbon. Rhizobium is a chemoheterotroph, which means it gets energy and nutrients from preformed organic energy sources, which have been synthesized by other organisms. Rhizobia that exist in free-living forms in soil feed on dead organic material and use it for growth and to provide substrates that are oxidized in cellular respiration to generate ATP. When rhizobia form symbiotic relationships with legumes, the photosynthetic hosts provide them with carbohydrates and all the necessary nutrients except nitrogen, which rhizobium obtain from the atmosphere. Even within individual species of rhizobium, specific strains can either be mutualistic, parasitic, or non-symbiotic. Mutualistic rhizobia provide hosts with lots of fixed nitrogen. Parasitic rhizobia rely on hosts, but fix little to no nitrogen to provide to their hosts. Non-symbiotic rhizobia only exist in free-living forms in soil and are unable to infect legumes at all. Legumes favor more mutualistic rhizobia. Mutualistic and parasitic rhizobia can produce millions of descendants. However, the rate of viability among said offspring is very low. All in all, non-symbiotic rhizobia often have more successful rates of reproduction. So here is an in-depth look step-by-step at the relationship between rhizobia bacteria and legumes. The process of root formation is initiated when a legume releases a chemical including amino acids, dicarboxylic acids, and low concentrations of excreted components called flavonoids through their roots, which the rhizobia in the soil are attracted to. Rhizobium accumulate near the root hair and eventually attach to the root hair's cell wall. Chemicals called nodulation factors are released from the rhizobium cells, which cause the root hair to curl. Now the rhizobium cells are able to digest the cell wall of the root hair and enter into the root hair. Rhizobium enters the root hair through invagination, which is the process of the cell membrane of the root hair being turned inside out or folded back on itself to form a cavity. The rhizobium bacteria enter the root through this formed cavity, which is termed the infection thread. Spontaneously, the cells of the pericycle layer of the cortex of the root become dedifferentiated, which leads to the formation of masses of undifferentiated cells called nodule primordia. The nodule primordia divide to produce outgrowths called root nodules. The rhizobia in the root nodule cells undergo physiological and structural transformation and are now called bacterioids. So the bacterioids are irregular shaped, non-flagellated, branched, and non-dividing forms of rhizobium. They produce the enzyme nitrogenase. Nitrogenase is responsible for the actual fixation of nitrogen into ammonia. This enzyme is very sensitive to the presence of oxygen and will actually become inactive when exposed to any oxygen. To avoid this, the bacterioids and the legume cells work to form a pink pigment, leg hemoglobin, that is an oxygen scavenger that removes any excess oxygen from the nodule cells. So that's how that works. The symbiotic relationship between legumes and rhizobia fuels the practice of crop rotation, which is used in many agricultural traditions. Crop rotation with legumes allows farmers to nourish plants who can in turn nourish the soil in a process that uses a biological catalyst at room temperature and ordinary pressures and uses sunlight to drive it. Farmers have been using legumes in agriculture since the ancient world to rotate crops to assure the fertility of their soils. In the New World, farmers use the common bean. In the Mediterranean, they use lupins. In China, they use soybean crops. In South Asia and Africa, cowpea was used. Crop rotation with legumes takes the place of relying on industrial nitrogen fertilizers, which is in the next slide. Overuse of nitrogen fertilizers can lead to the release of greenhouse gases like nitrous oxide and the eutrophication of waterways. Nitrous oxide's warming potential is around 300 times greater than that of carbon dioxide. Eutrophication is unwanted fertilization of a waterway that can lead to overgrowth of ecologically harmful microorganisms like algae and cyanobacteria, and eventually lead to the loss of all other aquatic life. The use of legumes that are symbiotically associated with rhizobia, which are naturally occurring soil nitrogen fixing bacteria in crop rotation practices, may help reduce the amount of industrially fixed nitrogen that is used, and that leads to loss of nitrogen into our atmosphere and waterways. So in conclusion, here's a picture of my back garden bed. I started growing some peas recently and have been very pleased to learn about rhizobia, which will hopefully continue to be utilized in the future, and on a more broad scale as a sustainable way to optimize agricultural food output and reduce reliance on more environmentally harmful agricultural practices like industrially fixed nitrogen fertilizers. This microbe has a lot of potential. Thank you for listening to my presentation. Here are some references for you. And thanks again.
My presentation is on spirochetes. The first image that I have on here is my portrait of the spirochetes, which includes the pieces of spirochetes as well as the Treponema pallidum, which is a specific spirochete that causes syphilis in humans. Spirochetes are gram-negative bacteria. The specific Treponema pallidum spirochete is a part of the spirochetes family. And in this image, it shows the specific pieces of spirochetes that include flagella, periplasmic space, outer membrane, the cell wall, and the protoplasmic cylinder. The image below is a zoomed in version of the pieces of a spirochete, which include the outer membrane, the flagella, the peptidoglycan cell wall, which is thin because this is a gram-negative bacteria, and the cytoplasmic membrane. The anatomy of spirochetes is very complex. They are gram-negative bacteria. They are helical shaped. They have a flexible outer sheath, periplasmic flagella within the periplasmic space, an inner cytoplasmic membrane, and flagella as well. The outer sheath is known as the outer envelope, and it is what provides structural support to the cell. The periplasmic space contains gel-like substances that hold the flagella. The flagella are also known as endoflagella or axial filaments, and they're what is around the body of these spirochetes in a helical formation, and they are not directly exposed to the environment. Down below is the picture of these spirochetes. The cytoplasm within spirochetes contains all of the DNA, ribosomes, enzymes, and everything that is needed for the bacteria to reproduce and metabolize itself. Spirochetes range in size because they are such a broad bacteria species. The flagella also helps them move. With the anatomy and the arrangement of this bacteria, it's able to move in a spiral motion. And since there are so many species and genus involved with spirochetes, some of them differ than others, but this is the basic anatomy of spirochetes, which is mostly the same. There are three main families of spirochetes. The first family includes the Borrelia, Brevinema, Cristospira, Spirochaeta, Spironema, and Treptonema. The second family is the Brachyspiraceae, which includes the Brachyspira. And the third family is the Leptospiraceae, which contains the species of Leptonema and Leptospira. Spirochetes are mostly known for causing syphilis. The Brachyspiraceae are mainly known for Lyme disease, and the Leptospiraceae are known for causing leptospirosis. There are many different species and genuses involved with them, and they can cause different bacterial infections and diseases within humans. The metabolism of spirochetes, they are very diverse with their growth patterns and metabolic capabilities. They typically thrive in aerobic environments such as mud and water. Some are strictly anaerobic, while others can be microaerophilic and can tolerate some oxygen. Since spirochetes are a very broad genus of bacteria, there are many different metabolisms and nutritional needs for different strains. Some are strictly anaerobic, while others can be microaerophilic and tolerate some oxygen as well. There are different ways that they can metabolize and different metabolic pathways that include glycolysis, the TCA cycle, and some of them even do fermentation. They have many different relationships with organisms. They can be mutualistic, pathogenic, and competitive. With the mutualistic relationships they have with different animals, such as cows and other organisms, where they can help with the digestive system and break down complex carbohydrates. They can also be pathogenic, which they can be harmful to humans and cause different diseases, such as syphilis and Lyme disease. So they can be harmful to humans and cause disease, but they do not cause disease in animals, as far as we know. And they can also be very competitive with their environment and other organisms and to be able to survive. And they will compete for nutrients. They can also have synergistic relationships with organisms where they can contribute to their environment, structure, and function of biofilms as well. This is a specific type of spirochete that I mentioned at the beginning called Treponema pallidum. It has a specific anatomy and it is gram negative. They are helically coiled, corkscrew shaped. It has a thin peptidoglycan cell wall and it is surrounded by an outer membrane that has lipopolysaccharides. There is multiple stages with this bacteria that include the primary, secondary, a latent period, and tertiary stages. These can all have different manifestations that present in humans and then cause many different symptoms within these stages. During the first stage is the early stage where you might notice sores of the infection where it entered the body. During the second stage, you might develop a rash which may be on the hands and feet. And during the third stage, which can also be the latent period, you may not see any signs, but the infection can live inside us for years. During the last stage, which is the tertiary stage, which most people do not get to, it can start affecting multiple organ systems that include the heart, brain, and nervous system. This can cause damage to many different organs and even cause death. If it does infect the nervous system, it can cause neurosyphilis, and that can cause many different symptoms as well. And there is a treatment for syphilis, which includes antibiotics that can help cure syphilis. However, you can still get reinfected with syphilis multiple times. And overall, spirochetes are very diverse, and there are many different families of spirochetes that can cause many different diseases within humans. And there are many different relationships that this bacteria has with people, animals, and its environment that may either be helpful or harmful. The way the bacteria metabolizes depends on what strain of bacteria it is, and the unique shape of this bacteria helps able it to move around its host and environments that allow them to survive.
cutie bacteria. Oh hey, it's me, Propionibacterium, or as some of you may know me, Cutibacterium. I thought I'd make an appearance so you can get to know me, as it seems I've been getting a bad rep because, well, I'm not in fact cute at all. I mean, I think I'm cute, but anyways, moving on. So I guess I'll start with the basics. I'm a pleomorphic, although generally bacillus-shaped and sometimes branched, skin commensal bacterium. Some other things you might like to know about me are that I am generally non-pathogenic and I'm mostly made up of common body fluids and blood. Pretty gross, right? Some other important factors are that I belong to the Coroniformes Actinobacteria phylum. I am also a gram-positive, non-spore-leading, aerotolerant anaerobic bacterium. I get my name because I have the ability to produce propionic acid through anaerobic catabolism. An interesting fact is that there is a dairy species that also comes from the Actinobacter phylum. So I have those distant relatives, and I also have closer relatives that are my same species. Their names are C. avidum, C. granulosum, and C. humorusi. I'm pretty well studied because I contribute to acne, mostly found on the face and upper trunk, but have been found in other areas of the body like the mouth, gastrointestinal tract, and groin. Although I am a normal part of the human microbiome, I can also cause other infections like endocarditis and post-operative infections. So like I said, I'm anaerobic, but I have genes that encode enzymes that enable me to survive in microaerophilic environments. I'm also part protein and have lipases that help me break down lipids and sebaceous follicles that provide me with energy. Whew, we have covered a lot, but there's still more. To recap, I thrive in low oxygen environments. While in these environments, with my species, we feed off sebaceous matter and produce molecules that help with inflammation. Then comes the not so cute part, where this causes acne, cysts, pustules, and papules, like so. Okay, I know it looks bad, but I'm trying to help. You see, the environment I like is also occupied by other pathogenic microbes that like to colonize there. I guess I'm kind of like Superman since I produce inhibitory properties that work against those pathogenic microbes. I guess I understand if you're still worried about me. I mean, my close relatives and I can cause infections, especially when I'm given the opportunity. This diagram breaks this down for you. Here you go. The good news is those infections can be treated. Treatment type and duration usually depends where the infection is on the body, but usually my isolates are susceptible to penicillins, cephalosporins, carbapenems, and clindamycin. Well, I really hope you enjoyed learning all about me. As for me, I have to go because I don't belong out here. Bye!
Tuberculosis Tales, Mycobacterium's Whispered Trails. I'm the cause of tuberculosis, TB, which the World Health Organization, WHO, classified as a deadly respiratory disease after I threatened the world in 1993. In fact, I'm so famous that the WHO publishes epidemic reports all about me every year. The 2019 report estimated that 10 million people were infected and around 1.4 million people suffered mortality. According to some models, the COVID-19 epidemic may worsen TB burden due to undernutrition, multi-drug resistant TB strains, unemployment, and redirection of resources to fight COVID-19. Who am I? Mycobacterium tuberculosis. I am a gram-positive, acid-fast bacillus with a very tough cell wall composed of lipopolysaccharide, mycolic acids, arabinogalactan, a polymer of arabinose and galactose, a thick peptidoglycan layer, and a lipid bilayer. Over 60% of my cell wall is made of mycolic acids, which contribute to my virulence. Other lipids in my cell wall, such as sulfolipids, cholesterol, and lipoarabinomannan, also help to increase my virulence. They help me be resistant to antibiotics, resistant to osmotic lysis, and oxidative stress. I hail from the bacteria kingdom, Actinobacteria phylum and class, Actinomycetales order, Mycobacteriaceae family, Mycobacterium genus, and tuberculosis species. I have several other relatives that form what is called the tuberculosis complex, because we all cause tuberculosis disease in either humans or animals. They are M. africanum, M. bovis, M. mungi, M. canneti, M. origus, M. microti, M. caprae, and M. pinnipedi. One of my relatives causes leprosy, and their name is Mycobacterium lepra. Not all of us cause tuberculosis, though, such as Mycobacterium smegmatis and Mycobacterium fortuitum, but they can cause human infections. Humans are my only known host, and I am spread primarily through respiratory droplets, such as sneezing or coughing. I am famous for invading the alveolar sacs of the lungs, but also invade the lymph nodes, bones, joints, central nervous system, heart pericardium, or bloodstream. I cause humans to cough, have chest pain, lose weight, fever, and even night sweats. If in the lymph nodes, the person can get abscesses. If in the central nervous system, I get quite a headache. My metabolism is heavily dependent on lipids, which are my primary carbon and energy sources. I'm such a chemoheterotroph. I steal lipids from my host because they are delicious, but I can also make my own. In fact, I can use cholesterol as my sole carbon source. Important enzymes responsible for fatty acid biosynthesis are FAS1 and FAS2, which produce some mycolic acids that make up part of my cell wall, such as PDIM, sulfolipids, polyacyl trehalose, and diacyl trehalose. Scientists predict that under aerobic and early growth conditions, I use glucose and triglycerides as carbon and energy sources, but then switch to using lipids once inside host macrophages where I cannot access glucose. Take that, macrophages, I can still replicate. Sometimes, a bunch of macrophages will chase me down and form granulomas by clustering together in large groups of foamy cells which starve me of oxygen. I must change up my metabolism if I want to survive, so I alter my Krebs cycle to favor succinate production. Succinate feeds succinate dehydrogenase, which is involved in both the Krebs cycle and electron transport chain. This means I still get to make ATP. To increase the success of my invasion, I must reduce the population of my enemies. Studies in TB patients have shown that I am associated with a decrease in microbiome diversity, especially short-chain fatty acid or SCFA-producing bacteria such as Firmicutes and Bacteroides. You see, SCFAs, such as butyrate, are helpful in immunomodulation, meaning that they help stimulate lymphocytes and phagocytes. I don't want that. Gut and lung dysbioses are my friends. I don't dislike all microbes in the human microbiome, though. I do like proteobacteria, so they are overrepresented during MTB infection. During anaerobic growth, they produce mixed acids, including succinate, lactate, formate, but also dreaded SCFAs. However, as I mentioned before, I do like succinate for my own ATP production. I also like some actinobacteria, such as from the genus Collinsella. No one knows exactly why these groups are overrepresented, and I'm not telling. I have many clever ways to escape the human immune system. One way is by purposely allowing myself to be internalized by alveolar macrophages, but then preventing the maturation of the phagosome. You see, during normal phagocytosis, the phagosome fuses with the lysosome to form a phagolysosome, which contains an acidic environment and enzymes that can kill me. One way I prevent lysosomes from killing me is by secreting a lysosomotropic, can penetrate lysosomes lipid called 1-TBAD, which functions as an antacid. Another way I do it is by secreting a protein, tyrosine phosphatase, PTPA, which prevents the ATPase in macrophages from doing its job of actively pumping protons and acidifying the phagosomal environment. Since I can damage phagosomes with my ESX1 effector proteins, macrophages try to come and repair them via the endosomal sorting complex required for transport, or ESCRT, and Gal3 proteins. However, my handy effectors ESXH and ESXG interfere with the recruitment of ESCRT. This prevents maturation into a phagolysosome and thus keeps the pH high enough to keep me comfy. Humans have found that ESXH and ESXG complex with each other and prevent ESCRT recruitment, thus disrupting its function, which also includes moving me to lysosomes. Gal3 gets recruited to damage phagosomes when internal glycoproteins get exposed, but my vectors block that signaling too.
My favorite microbe is Helicobacter pylori. H. pylori is gram-negative, corkscrew-shaped, and flagellated. It is a bacterium of the Epsilon proteobacteria class, and its circular DNA is housed within the single-celled organism. Its ability to survive in such a harsh environment would suggest that it is an extreme acidophile, but it is a neutrophile. H. pylori can be found adhering in the stomach and duodenum. Diseases H. pylori contributes to are often mistaken for presence of the microbe itself. However, H. pylori can be found in the stomach of over half of the world's population. The microbe's presence is not the issue, it is the chronic colonization and infection that becomes an issue because it can lead to disease. A majority of those with chronic H. pylori will experience ulcers, gastritis or duodenitis, and a smaller portion may develop stomach cancer or stomach lymphoma. Two of the most useful components on the surface of the organism are lipopolysaccharides and urease. Lipopolysaccharides assist it in adhering to other cells, while the enzyme urease converts urea and water to carbon dioxide and ammonia. After H. pylori has colonized, it begins to eat its way to the epithelial cells of the stomach. When the previously mentioned enzyme urease turns the acidic urea into carbon dioxide and alkaline ammonia, it creates a neutral environment that allows it to survive. The flagella of the bacteria help guide it further toward the stomach's epithelial cells to target its tight junctions. Once it makes contact with the cells, it adheres to them with the assistance of lipopolysaccharides, and then it begins to excrete exotoxins. These exotoxins are known as vacuolating toxin and cytotoxin-associated protein. Kage disrupts cell integrity and breaks down the tight junctions between the stomach cells. Kage also stimulates production of Illinois-8 that attracts neutrophils that cause inflammation. VOCA induces apoptosis of stomach cells. VOCA induces apoptosis of stomach cells. This allows the acid layer and mucus layer to come inside of the space H. pylori has created. As the layers come in, the acid layer damages the tissue and forms stomach ulcers. Inflammation also promotes more hydrochloride secretion in the stomach because of histamine. This volume increase allows acid to enter the duodenum, which is usually basic itself and that can cause duodenal ulcers. There are treatments available to rid the host of H. pylori, including proton pump inhibitors to rid the stomach of acid, bismuth to kill bacteria, and antibiotics to kill the H. pylori.